Good afternoon. I'm Jim Hurley. I'm a structural biologist in the NIDDK, and I'd like to welcome you to today's Wednesday afternoon lecture by Dr. Alfred Widdinghofer, who we all call Fred, who's a structural biologist who hails from Dortmund, Germany. And Fred was just telling me that his PhD was actually on a subject of interest historically in our institute. He participated when he was a student at the Wohlforschung Institute in Aachen, Germany, on the first chemical synthesis of insulin, and his role in the project was to put protecting groups on the A chain such that it would efficiently form the correct disulfide bonded dimers with the B chain without giving rise to irrelevant mixtures. So Fred, I think, became known to many of us in the late 1980s when he solved the correct structure of the RAS small G protein, which really was the archetype for the signaling group of G proteins, and ever since then has been at the forefront of G protein research. And I really can't think of anybody who's contributed more to the understanding of this extremely important class of proteins than Fred. In 1993, he became director of the Max Planck Institute for Structural Biology in Dortmund, and I recently heard that he was retiring from that position. He's stepping down in June, and fortunately, he's going to continue to have a lab for a number of years. But when I had heard that he was retiring, one of the first things I did was nominate him for this lecture series, because I really thought we'd all wanted to get a chance to hear him and meet him, for those of you who haven't, before that happened. In terms of extracurricular interests, he's a big supporter of the Borussia football team in Dortmund. So his second office, I guess, is in the stadium there. I first met Fred, I think, in 1995 when he came to give a talk at a symposium at NIH, and as just a starting young investigator, and I had a poster on our structure of the C1 domain of protein kinase C, which I just solved and was struggling to publish at that point, and he was the first real leader in structural biology who noticed that work and realized its significance. So I really appreciated him for that, and we've crossed paths in a number of interesting ways over the years. I think the next time I saw him was in Israel at a conference, I think in '98, and Zvi Zellinger took both of us on a tour to the Dead Sea, and we had a photo op swimming on our backs and both reading Science magazine, and unfortunately neither of us scanned the picture in time to bring it, unless Fred's going to surprise me. So I'm probably eating up too much of his time. I should not omit to mention he's won numerous awards. He's won the Louis Janté Prize for Medicine, the German Cancer Prize, the Otto Warburg Medal of the German Biochemical Society, and many other things, and we're looking forward to a great talk.
Is the microphone working? Is it working now? Okay. Thank you, Jim, very much for the honor to speak here. Since he mentioned retirement, obviously if you have these kind of talks, keynote lectures or prizes or whatever, it tells me that I'm probably getting too old, because this is probably how you can determine the age of a scientist. This is when you're young, you work a lot, and later when you get him, so this is about my age, when you not work much, you travel a lot, sit in too many committees, and give keynote lectures. So what I would like to do, I mean, what has been sort of accompanied all my life, is to understand a major problem in biology, not problem, but a thing that drives a lot of biological reactions and signal transduction pathways, and that is that the addition of a phosphate to a protein changes dramatically sometimes, or very often, the properties such that it has now a different orientation, a different activity, a different localization, different interaction partners, and so on. And the way it normally works is that a protein gets phosphorylated by a kinase, and then it has a certain effect, a new effect that it didn't have before, and then a phosphatase cleaves it back into the old stage. And the point charge of a phosphate is something that is really very, very interesting in terms of it is kinetically unstable in terms that it never hydrolyzes, but thermodynamically it's a downhill reaction, but it never happens, which means that removing phosphates is always a process that requires an enzyme. And there is an interesting review that I recommend to my students always, written by Francis Westheimer, one of the great old guys of phosphor biochemistry, which says, why do chemists never use phosphates as leaving groups, whereas lots of biology is driven by this process. And this has to do with the kinetic stability of a phosphate bond. But the protein that we are working with work by a similar principle, except here that it is not the protein that becomes phosphorylated, but rather the nucleotide that is bound to the protein. But the difference again between the one state, usually the off state, is when it has GDP-bound, and it is on when it has GTP-bound. So the difference is just, again, a single phosphate. And the way these proteins work is that they bind GDP very tightly. That means he never dissociates GDP. It needs a factor, it's called guanine nucleotide exchange factors, that drives GDP out and allows GTP to bind. In a GTP-bound form now with the extra phosphate, it can now interact with lots of other proteins. And then there is a GTPase reaction, which is again very slow and activated by proteins called GTPase-activating protein. People usually call these proteins a GTPase. But it's nothing else than a GTPase, because this reaction, let's say in RAS, is 10 to the minus 4 per second. This reaction is 10 to the minus 4 per second. So the whole cycle of substrate binding and hydrolysis is 10 to the minus 8 per second, which is much slower than GTP hydrolysis in water. So I think GTPase, even though it's a nice short name for it, is a misnomer. I will be talking a little bit about this here, and mostly about the GTPase reaction, how it affects many diseases and things. So it is an on-off switch that in this usually form is off, and in this form it's on. And there are about 300 or so different G proteins or more. I mean, so with isoforms. And they all have this structure, at least the one domain that we call the G domain, which is the domain that binds nucleotide, that hydrolyzes nucleotide, that has this conformation change from the GDP to the GTP-bound form. They all have conserved sequence elements indicated here, not surprisingly, around the nucleotide binding site. And then they have a three-layered structure, as shown here, a beta sheet in the middle and helices on both sides. So that's how they all look, but they have very many different functions and very many different sort of properties that I will elucidate while we go along. And first of all, in the first part of my talk, I will talk about this superfamily of what is called RAS-related proteins, which all have sort of 20 to 25 kilodalton. The founding father, the grandfather of the whole family, is RAS itself, the first of the small G proteins to be isolated, and all the others were discovered later on, and they were actually named after RAS. For example, Rho stands for RAS homolog, or RAT stands for RAS in diabetes, RAN stands for RAS in the nucleus, and so on. So all the names derive from the godfather of the family, and we in the lab have been working over the years with many of these, and I will briefly mention RAS. We'll be mentioning RAP, REB, and AL here, and I will explain that while we go along. So for a structural biologist, obviously, one of the most important questions in the beginning, when we started working on this, is a long time ago, 20 years or almost ago, I think, or more, was the question, how does a single phosphate make this conformation change that drives this biological reaction? And it's actually a very simple principle. How these proteins work, and actually many ATP-binding proteins work in a similar way. And that is that the major part of the structure doesn't change when there is GDP or GTP, and there are two areas which are called switch 1 and switch 2, which have a totally conserved residue, threonine or glycine, which is the numbers here are coming from RAS. They're bound to the gamma phosphate by 2 main chain hydrogen bonds. And then they keep that in a fixed conformation. But once you relieve the gamma phosphate here by the GTPase reaction, these mobile regions relax into a different conformation. And that is the basis of the whole molecular switch of how they all work. They all have the same switching mechanism. And I'll show you just a few examples. This would be RAS, and here down here would be the switch areas. This would be the gamma phosphate, and the main sort of conformation changes down here. And all the proteins that interact with RAS recognize this area down here. A similar change, but much more dramatic, is, for example, the protein run. Again, the canonical switches down here, the release of gamma or the binding of gamma phosphate makes this large conformational change. And just another example, for example, here, which is the protein ARF or AL, and again, which is a very dramatic change in that these two beta strands of that beta sheet changed by 2 amino acids along the rest of the beta sheet, just when an extra phosphate is bound to the protein. Very dramatic changes, but they're triggered always by the same kind of interaction, yes or no, with the gamma phosphate. So before I start to talk about GTPase, I will briefly summarize another important element of how these proteins work, and that is effector binding. So they, in the GTP-bound form, they recognize proteins, or are recognized by proteins that are called effectors, because they mediate the biological effect of the particular protein. And we have solved, and other people have solved, a large number of structures. For example, this would be RAS, how it binds to the RAS binding domain of the RAF kinase or of another downstream effector of RAS, doesn't really matter. What I would like to show you here is that what they recognize is these blue areas of the protein, which is the switches, because these switches are the ones that change conformation when there is a gamma phosphate, yes or no. And not surprisingly then, all the effectors somehow recognize that. So if I would overlay all the effectors that have been determined over the years, you would see there's lots of spaghetti. Obviously, there seems to be no order in it. And I try to order it according to the principle of what these effectors here, just these were the ones, most of the ones were done by us. They recognize basically at least also at either switch 1 or switch 2. So all the effectors that have been known over the years, whose structures have been determined in complex with the RAS protein or RUN protein or whatever, Rho, they see whether the gamma phosphate is bound or not by binding to the switch areas. For example, an important effector of RAS is the RAF kinase, for example. I mean, there are about 1,000 papers on RAF kinase, and we still don't understand how it functions. But what we know is that the RAS binding domain called RBD, which is in the regulatory part of the RAF kinase, binds to RAS when RAS becomes activated. And even though we don't understand sort of the mechanism, what we do know is that this protein gets recruited to the plasma membrane where RAS is incorporated once RAS becomes activated. And that's why these RAS binding domains are very great tools for studying in vivo the activation of RAS proteins. And I'll show you here sort of an example where we labeled with GFP RAS and the RAF RBD. And when I now hit my computer and hit the mouse, what I will actually be doing in the experiment that I'm showing here is an EGF. I'm treating the cells with EGF, and you see what happens. This is a state at 0 point times 0, and I try to stop it as fast as I can. And this would be the starting situation here, and this would be the situation after 4 or 5 minutes. I couldn't stop it that fast. So you see there is RAS is in the plasma membrane, and the RAF RBD is in the cytoplasm. And in a few minutes after I hit the cells with EGF, there is a series of reactions which in the end recruit the cytoplasmic RAF RBD to the plasma membrane and later on to the Golgi. So these RAS binding domains, which specifically recognize the GTP-bound form of RAS, are very important tools to study in vivo signaling of RAS or Rho or RUN or whatever. RUN is not a signaling, but RAS or Rho and its isoforms. But I would like to sort of more concentrate, what I've told you is that the G domain is sort of a conserved switch module, and I've shown you some of the variations. I show you that effectors bind to the GTP-bound form, and that actually the effectors, the very sense of signaling via RAS or Rho and the others is that once they become activated, they recruit and or activate effectors, for example, the RAF kinase. But I would like to concentrate more on something that we have been following over the years in many, many cases, and that is how the inability to hydrolyze GTP leads to many different diseases. And obviously the classical case is RAS itself, which, as you know, is also like a switch here, and it's an oncogene, as you also know. It is mutated in about 20% of human tumors, and the effect of that is that it cannot hydrolyze GTP anymore. And what that leads to is that RAS accumulates in the GTP-bound form because it cannot hydrolyze anymore. And so you don't need any of the upstream signaling stuff anymore. RAS is GTP-bound all the time and has its downstream effect all the time, and that's how it contributes to cancer formation. And the reason I still tell this story, even though it's about more than 10 years old, is sort of a paradigm for understanding the reaction, is that a textbook that many of you might read, very famous textbook, says exactly how GAP interacts with RAS and perhaps other components of the RTK-RAS pathway is unclear. I already showed you how RAS interacts with RAF kinase, and I also show you how it interacts with GAP. So this really pissed us off quite a bit, and or maybe I shouldn't say that, this is the president's special dinner speech here. But anyway, we were a bit mad about it and sent these people a bunch of papers, what we thought were prominent papers, and then apologized, said in the next version of the textbook in about 4 years they would correct this. Hopefully we see that then corrected. I briefly, really, because it's an old paradigm, I briefly recapitulated before going to some newer stuff. So all these gaps are specific for the particular subtype of RAS protein. So there are about 13 or more gaps that are specific, more or less specific for RAS itself. And they all have, and you see they have many domains, but they all have a RAS gap domain of about 300 and so amino acids, which is sufficient to hydrolyze GTP. Because, as I told you, the hydrolysis reaction is very slow, 10 to the minus 4 per second, and gets stimulated by these gaps about 10 to the 5-fold. And it's not at all stimulated if you have oncogenic RAS, which has a point mutation in either of two positions. And the way, and I come back to this scheme, that's why I show it in a little detail. What we always do is biochemistry, sort of kinetic studies and structure and so on, which are in the end combined. So what we do, for example, we do a stop flow kinetics. So we shoot together RAS GTP, which has a fluorescent group. We shoot together with RAS gap in a mixing chamber. It's called stop flow because the flow is stopped by a stopper here. And then we watch the reaction. This is the fluorescent derivative of GTP. And if you shoot these two things together, RAS man GTP, one of the RAS gaps is called neurofibromine, and it's the domain because it's 333, that's the size of the domain. So what you see here in green is increase of the fluorescence and a decrease, which means it binds to the gap and is hydrolyzed and it dissociates and it's all over in 1 second, where normally it would take hours for it to ever hydrolyze. And what we also showed is that there is an arginine in these gaps. If you mutate that to anything else, then you get binding all right, but no hydrolysis, and we have called this the arginine finger. And what is also important for you to remember is that many phosphotransfer enzymes use aluminium fluoride in the active site they sit in the gamma phosphate binding site of, let's say, RAS GDP, but only and other phosphotransfer enzymes, but not as a ground state homolog, but as something that mimics the transition state of the phosphotransfer reaction. Because it is a flat trigonal aluminium fluoride thing with a nucleophile, incoming nucleophile on this side and the leaving group on this side. And all the RAS proteins don't do that. It's only when they have gap, when they have a 1-to-1 molar complex between RAS and RAS gap that they bind aluminium fluoride, which was for us an indication that RAS are incomplete enzymes, incomplete phosphotransfer enzymes that need the presence of a gap to make this complex and mimic the transition state of the phosphotransfer reaction. And indeed, that's how we were able then to finally solve the structure of RAS GDP with aluminium fluoride and GAP, where this is RAS GDP with aluminium fluoride, and the green stuff would be the GAP. And you see here, this would be the arginine that goes into the active site, stabilizes the transition state, and so on and so forth. So what we have here is an incomplete enzyme, or an enzyme that is formed by two proteins, and only when they come together do they form an efficient complex, an efficient enzyme. And indeed, one finds in these complexes always GDP aluminum fluoride to be being a flat pancake and the nucleophile being there and the leaving group being there. And what is also important and will be important in later cases is that there are two elements that are really crucial for the reaction. One is the glutamine. This is from RAS, and this is the arginine finger from GAP. And without going into any details, you can see that what glutamine, for example, is doing, it stabilizes the nucleophile. What the arginine is doing, it takes away the negative charge in the transition state and also stabilizes the glutamine. So these are then the crucial residues that are important for catalysis. And this, for example, is one of the residues that, when mutated in RAS, makes RAS an oncogene that can no longer hydrolyze GTP. And it's obviously clear that, let's say, if this is a leucine, for example, that this reaction scheme cannot be observed. And that's why there is absolutely no hydrolysis whatsoever. But to show you that this is important for many other diseases, so 25% of human cancers, or 20%, somewhere in between, can't hydrolyze GTP. But there are other diseases that also depend on RAS being unable to be hydrolyzed. One of which is type 1 neurofibromatosis, a tumor, which is a disease that shows benign tumors. This is here a mild form of the disease with these café au lait spots on the skin and small tumors, and they can be very, very disfiguring. And neurofibromin, as I told you, is a gap for RAS. And so when it is mutated in two copies, so it's a tumor suppressor gene whose two copies need to be deleted or mutated, then people get this disease. And we were sort of having, when we discovered these stories, there was a colleague from the Charité in Berlin came to us and he said he has a woman that has a disease, died at the age of 35, and her 3 sons have the disease also. And when we analyzed, in the blood she is heterozygous, and in the tumor she is homozygous for the deletion or mutation, and she has actually a mutation where the arginine is mutated to a proline. And obviously this is the arginine finger that is mutated in this particular patient. And when we do the analysis in the biochemical assay, you can see that this arginine mutation to Q or P, which is another, so two of these patients we discovered, they do bind to RAS, but there is no hydrolysis whatsoever. So there are two different reasons to be where the deletion or where the inability to hydrolyze RAS can lead to two different diseases. And that's why, even though I have only a few more years to go, what we're still having a dream is, can we develop small molecules that would maybe induce oncogenic RAS to hydrolyze GTP? And it's probably a wild dream. Everybody tells me when I go to drug companies, they say, that's great stuff, but please show us that it works. And so nobody helps us, and it's still something that we are following, that we think the chemistry of hydrolyzing oncogenic RAS should be fairly simple. We just need to find small molecules that find the surface of RAS and do hydrolysis there. And I think we hopefully will ever get there. But that's sort of the last thing that I want to do in the next 2-3 years. I just want to give you a few other examples of first of all, that there are completely different phosphotransfer mechanisms, and second of all, that these lead to different diseases in many, many, many cases, and I will show you 2 or 3 examples. For example, there is a protein called RAP, stands for RAS proximate. It's the closest homologue of RAS from the RAS subfamily of the superfamily of RAS proteins. It has a totally different sort of biological function as RAS. It's involved in integrin and platelet activation. And the reason it was interesting for us is that this is the only RAS-like protein that doesn't have a glutamine, which we thought is absolutely essential for catalysis, because, as I told you, in oncogenic RAS, it's mutated to any other residue, and hydrolysis is kaput. And furthermore, RAP gaps, so again, there are specific gaps for this protein called RAP. They have no arginine finger. So 2 of the absolutely crucial residues are not there. And we asked ourselves, how does this protein hydrolyze GTP? So again, there is a number of RAP gaps in the database, human RAP gaps, which are indicated here by this blue, dark and light blue domain. And why this particularly interesting is also that a tumor suppressor gene called tuberin, or actually TSC2, has the same domain and as we show, the same mechanism as these RAP gaps. And also, RAPGAP has been found in some of the sequencing efforts as a cancer gene. One of the isoforms of RAPGAP called SPAR1, its knockout in mice makes a CML-like phenotype, a myeloid leukemia phenotype. And also, it has been shown that germline polymorphism in SPAR1 seems to be associated with poor prognosis in breast cancer. So it seems to be involved in cancer-related sort of phenomena. And the tuberous sclerosis, where the tuberin is mutated, is a disease that has been called tuberous sclerosis because people in the brain have these sclerotic sort of tubules, and they are usually mentally retarded. That's how they actually are detected. But they also have tumors which are called adenoma sebaceous. And so it's, again, a benign tumor caused by the deletion or mutation of the GAP tuberin. And the pathway that is involved in is shown here is actually the insulin-like pathway whereby PI3 kinase becomes activated, activates AKT or PKB. This in turn activates or somehow inactivates the tumor suppressor TSC1 and TSC2, one of which is a GAP. And they are the GAP for a protein called REB, which activates mTOR, and that is involved in regulation of cell size and so on and so forth. And so tuberin is then here a negative regulator of REB, and any positive regulator of the pathway are oncogenes, and any negative regulator, like P10, for example, are tumor suppressor genes. So that's why it is a very important regulator of growth in people. So let's come back to the mechanism. And again, we did this stop flow experiment where we shoot together RAP now, MUNT GTP, with RAPGAP, and we get hydrolysis. This would be the wild type situation. And we did obviously a lot of studies to find out what is the residue that might be important for hydrolysis. And it turns out there is an asparagine. If we mutate that, we again get a complex formation, but absolutely no hydrolysis. I don't know where that is. In 5 kilometers from here, it would be still up there. So absolutely no hydrolysis when you mutate a single residue. And it doesn't affect binding. Binding is even better than wild type. So a single residue makes catalysis in this particular reaction. And the same thing can be shown by formation of an aluminum fluoride complex. It's again, wild type forms an aluminum fluoride complex, and the mutation doesn't do that. Again, a strong indication that this residue is important for catalysis. So no transition state if that mutation is in the protein. Now we solved the structure and identified sort of the catalytic site and all that. This has been done by a student that's called Olli Daumke. So we identified here this residue. So we made obviously many mutations and identified this residue in the end as being catalytic. If you mutate that, there is a 10 to the 5-fold reduction in GAP-mediated catalysis. And he called this, and I'm sure only German people understand this, and there are a few around here, that's why I tell this. So he didn't want to call it the Asenfinger, and that's what he called it, the Asendaumen, the thumb, because his name has a connotation to this here. So don't laugh, please. So that was obviously, we could identify the residue, but what is the mechanism? Why would such a single residue be so bad for this reaction? Completely wipe it out. And obviously, only structure can tell you that. I mean, you can do many biochemical studies, but in the end, you need to go to a structure that tells you how it all happens. And before I show you that structure, I should point out again that in tuberous sclerosis, so this is a sequence alignment of many RAP gaps, and this is tuberin here. So the protein that is whose deletion or mutation leads to tuberous sclerosis. And again, you see these mutations here, indicated here, and where it is conserved. For example, this is the most highly conserved region, which is the catalytic site here, and this is the asparagine, and it's mutated in again with patients with a disease. So again, a crucial catalytic residue leads to disease, tuberous sclerosis. And the structure shown here has been done by Andreas Grima. And the blue and green stuff is RAPGAP, and the red stuff is RAP. And you see what this arsen is doing is pointing again into the active site, just as before the arginine finger was pointing into the active site. So what would it be doing? Why is arginine pointing into the asparagine pointing into the active site a catalytic sort of residue? Answer is very simple. if we overlay, oh, something happened here, sorry. So if we overlay a number of structures, RAS and RAS gap, Rho and Rho gap, with RAP gap, we see that all of these structures have, this is the triphosphate, this would be the catalytic water. All of them have a glutamine that we know is important for catalyzing or stabilizing the position of the nucleophilic water. And here is RAP, which has a threonine in that position, points away from the active site. But now, RAP gap, this is the catalytic helix here, points this asparagine into the active site, just at exactly the point where you normally have the intrinsic glutamine. So RAP gap uses a carboxamide in trans, stabilize the water, and that alone stimulates hydrolysis by a factor of 10 to the 5. So a completely different mechanism as in RAS, but it's also important for disease mechanisms. And the last example for a disease mechanism is something that we found recently, and it was, again, a structure told us what the function was, because it turned out that we were working on proteins called ARL, and there are 16 members, and they're belonging to the ARF subfamily. ARFs are involved in forming vesicles, and some of the people here in the room I know work on this and know much more about it than I do. But these 16 ARL proteins, nobody really has a clue what they're doing, except for ARL2 and ARL3, which have some connotations to retinal diseases. Because ARL2 and 3, there's a gene called RP3, which is a retinitis pigmentosa gene product that interacts with PDE delta, a protein that interacts with RL2/3, is again a phosphodiesterase subunit which is found in the retina. RP2, another disease of the retina, is mutated, and it's supposed to be an RL effector. And another protein found specifically in the retina called HRG4, human retina gene 4, is also a protein that interacts with RL2/3. So this seems to be a very strong indication that somehow RL2/3 is probably involved in ciliary transport between the inner and the outer segment of the retina, of the human retina, of the photoreceptor cells of the retina. And this is how people with retinitis pigmentosa, this is how they see this building. They have a central view only, and the peripheral view is completely gone. And you have these pigments in the retina, that's what the name comes from. So this RP2 protein, retinitis pigmentosa 2, is coding for a certain isoform of retinitis pigmentosa, an X-linked form. And this was the structure that we solved. And it was supposed to be an RL effector, so a protein that interacts with the GTP-bound form of RL exclusively. So we solved the structure. There were a number of mutations, and most of them are involved in stabilizing the structure. You can easily see of how mutations of many of these residues would sort of destabilize the protein. But there are two residues, arginine 180 mutated to histidine, lysine, cysteine, which is pointing outside and unlikely to be a structural residue, but rather a functional residue. And so obviously we asked ourselves, what is it doing? And again, the clue came from an experiment that my student did, not really by chance, but we were surprised, because when he did the following experiment, he took RP2 and RL2 in a GDP-bound form and ran it over a column, a gel filtration column. Obviously, RP2 and RUN separate from each other because obviously they are not supposed to be binding to each other. But when he does the same experiment in the present aluminum fluoride, they suddenly elute together, form a complex. And that obviously rang a bell, because if a protein stabilizes aluminum fluoride binding, it's clear that must be a gap. And indeed, we solved the structure of the complex, and you see here, so this is the complex. The green is the RL protein. This is the beta helix of RP2. This is two domains of the protein. But what you see immediately, obviously, is here an arginine poking into the active site. So obviously the structure told us what this protein is. It's a gap for RL3. So retinitis pigmentosa is caused by mutations in the gap for this protein. And actually, if you look into the active site, GDP aluminum fluoride complex, all these residues that you see here are mutated into various other amino acids in the disease retinitis pigmentosa. So a structure really told us what the function of the protein is. So let me conclude before I come to the last sort of 15 minutes of my talk. So what I showed you is that RAS superfamily proteins are almost always incomplete enzymes, that have a very slow GTPase reaction, and they need GTPase-activating protein in order for to be hydrolyzed and to be regulated from a GTP to a GDP-bound state. And what GAPs usually do is they stabilize the switch and the catalytic glutamine if it's there. Many GAPs supply a catalytic arginine residue, what we call the arginine finger, and RAPGAP supplies an asparagine that does the same thing as the glutamine, intrinsic glutamine of RAS and Rho and AL and so on. So gluten is essential for catalysis except for RAP, where it is supplied in trans by somebody else. What I haven't shown you is that the GTPase reaction is reversible and so on and so forth. But what I also have shown you is that the deletion or mutation of GAPs can cause a variety of diseases, and there are many more examples, but these are the ones that we worked on. And we think that the GTPase reaction is a valuable, at least as in case of RAS, a valuable anti-cancer target. If you could ever induce oncogenic RAS to hydrolyze GTP, that is its primary defect, we think it would be a good lead into maybe an anti-cancer drug target. In the last sort of 50 minutes, I would like to show you something completely different, because so we have so far talked about proteins, monomeric small g proteins, GTP-binding proteins, that have this cycle here, which is regulated by GEFs and GAPs. And in the GTP-bound form, they bind to effectors and do the biological effect. RAS is growth, Rho is actin cytoskeleton reorganization, AL, we don't know what it is doing, and so on. But there are a large number of other G proteins, GTP-binding proteins, which work as a dimer, where usually GTP-binding is not very tight. They don't need an exchange factor for binding GTP. But what they always need for function is that these two monomers come together in the GTP-bound form. And usually their GTP binding is also connected to the biological effector reaction. That's why we call them G protein regulators. And then that induces a biological effect. For example, what you probably all know is signal recognition particle and its receptor, the two proteins that are involved in taking signal peptides to the ER membrane, to the sec translocation channel. This is, for example, such a case. And I will show you another case that we worked on recently. There are three types of these proteins that we call GTPase-activated by dimerization. Some of them do pseudo-homodimerization. So, for example, the signal recognition particle and its receptors, they are two different proteins, but yet their active sites are completely identical, and they stimulate each other's GTP hydrolysis, and by doing that, they also mediate transport of protein into the ER. I show you one little example about septins that we have worked on. Probably the transport into chloroplast by TOC proteins is probably also by a similar principle. Another case is homodimerization, where two monomers of the protein come together. Most famous example is dynamine, and we have worked on a few other of these. Dynamine also coming together in there with their active site probably stimulate each other's GTP hydrolysis. And there is homodimerization in cis, and I will show you one example that we've worked on recently, which is called the LARC protein kinase, which is mutated in patients with Parkinson's disease. So this is the common feature of all of these proteins activated by dimerization is, and these are three of them examples are from us, and this is the SRP-SR case, is that they come together across the nucleotide binding site and stimulate each other's hydrolysis, and at the same time also stimulate the biological reaction as a dimer. This, for example, is a structure that we recently published, and I was just in Janelia Park on a meeting on septins, and we determined, for example, the structure of the septin filaments, where you have also nucleotide binding across these interfaces here and here and here and here. And this is a G protein that forms long filaments whose really functions are not yet known, but it's a completely new type of filaments which are called septins. Another protein that we worked on, and which is, as an introduction to the Parkinson kinase, is a protein that is dimer already, but the two G domains come only together in the GTP-bound form, and then they're doing something that has to do with tRNA modification. But I just show you that only to make sure that you understand that this protein, although dimer, although already a dimer, but the G domains are far apart from each other in the GDP-bound form. You add GTP and then they come together. We just discovered this mechanism recently and forms this type of interaction which we call kiss of G domains. And the person who does this is my student Simon. So the last 10 minutes then is on LARC2, the Parkinson kinase. And it turned out, so it started when somebody, Peter van Hastert, in working on dictyostelium, discovered a huge protein called GBPC, which is a protein that binds cyclic GMP and has many other domains: a RAS exchange factor, domain, a kinase domain, a leucine-rich domain, and something that is called ROCK-core. And he called this, and later found that this tandem of two domains is found in many proteins. And he calls this ROCK for RAS of complex proteins, and core stands for C-terminal of ROCK. And they always come together in many proteins in dictyostelium. He discovered them in dictyostelium first, and then he found them that they're also in humans. There are 4 types of ROCK-core proteins, and one of which is the kinase that is mutated, LRK2, is a kinase mutated in Parkinson. And it has a LRR, a ROCK, and a core, and a kinase, and a WD40 motif. So we wanted to work on this. We wanted to understand how these tandem domains, ROCK and core, how they function and what they're doing, and how this is involved in Parkinson. What has been found about the Parkinson mutation is that all the mutations have a LARC kinase that has a higher activity. So the disease is caused by a higher kinase activity, but you find mutations in LRR, in ROCK, in core, in kinase, and W everywhere, but they all lead to an activated kinase. The problem was, obviously, as a structural biologist, you have to express proteins or domains of it, and all of them were insoluble. So we couldn't work on them. That's why we, at least to understand the function of ROCK-core, we turned to a number of prokaryotes that also have this ROCK-core LRR domain combination, and those were soluble, and we could at least, and I show you that working with these bacterial proteins told us a lot about, we think told us a lot about the kinase in people. And this is the work of Katja Gotthard, a student who just finishes up. So this would be, again, the structures. It would be the mutations that you find in patients. You see they're distributed all over the protein. So what we did, we obviously solved the structure first of the core protein. It is a dimer. You see that here. This is one protomer. This is another one. This is the N-terminal domain. This is the C-terminal domain. And we saw that, for example, this is a residue that is conserved between prokaryotes and people. But obviously the structure didn't show us anything. This is a tyrosine, points into solution, so what? If it's mutated to anything else, shouldn't make any damn difference. So that didn't tell us anything on how this, on the function of such a thing. But what was really sort of interesting when we started to make the structure of the ROCK-core tandem. So this would be, again, core, the dimer here that is dimerized over here. This would be the G domain connected to this core. And we found only one ROCK, but it doesn't really matter from what the conclusion that we can draw about it. So this is a typical RAS protein here, very much aligns with RAS, connected to the core domain. This is a dimer. So if we now look, and this is just showing that it is like RAS, but if we now compare, and obviously what a structural biologist always does is when he solves the structure and asks the question, what is the homology between all the different ROCK-core proteins, what is conserved? And if you do that, and look at the conserved residues, for example, this would be a core as a ribbon, and ROCK in van der Waals representation, and the coloring would be conservation of residues. And you see, red means totally conserved between the prokaryote and the human LARC kinase. And if you do the reverse experiment, you look at the van der Waals surface of the core and look at RAS, the ROCK domain in ribbon, and again, the conserved residues on core are red here, again at the interface. In interface between ROCK and core, you find residues that are totally conserved between prokaryotes and the human kinase. And so you ask yourself, so what do these residues do? Let's first look, for example, at, so this is where these residues are located, in the interface. This would be switch 2. Remember, switch 2 is one residue that changes conformation when it goes to when the gamma phosphate is there or not. It's a very flexible region here, but it sits also in the interface. And for example, some of the residues, for example, this tyrosine here is totally conserved. This tyrosine here is partially conserved. But all the mutations in people, at least the mutation in ROC or core, are in that interface between core and ROC. So in other words, there is something in the interplay between these two domains that is involved in the disease when it's not properly functioning. And it turns out if you make mutations in that prokaryotic protein and ask the question, what happens? So this would be the GTPase reaction of the wild-type protein, and this would be the mutations that are analogous or even identical to Parkinson. You see that they all reduce the GTPase reaction. And what is also clear is that dimerization is required for the GTPase, because if you now take a mutation that has, where you wipe out the dimerization interface of the core domain, so this would be, again, the wild-type GTPase reaction. This would be the one where you prevent dimerization. So in other words, you need the dimerization for an efficient GTPase. And that's why sort of we conclude from this that somehow there is these ROC domains have, in the GTP-bound form, they have to come together to stimulate the kinase domain, because what we all know is that all the mutations in LRKK2 are increasing the kinase activity. And I think if the GTPase reaction is reduced or slower, you have a higher kinase activity, because somehow this interaction between the two GTPase domains induces kinase activation, probably by dimerization, because as you know, many kinases need to transactivate each other by phosphorylation of the activation loop. So that would be our model that we're now testing, trying to test at least in humans. And from all the evidence that is around, we think we can explain by this mechanism that there is a dimerization of G domains, which somehow is transferred to the kinase domain, allows them to autophosphorylate each other and thereby activate their kinase. And this is analogous to RAS, because if RAS cannot hydrolyze GTP, it also has a longer time to interact with RAF kinase, increases the RAF kinase activity, and that leads to the disease cancer. And here it would lead to disease Parkinson, even though we don't know what the LAR kinase is doing. But we know it is a higher activity in the patients. So let me sum up then and mention the people that have been doing the work. I've shown the pictures all along. RAPGAP, that was done by Olli Daunke and Andreas Kriemer. That's why he called this the Asen-Daumen. Septins, I briefly showed it only, is done by Mina, who is now in Ron Wales' lab, and by Marian Farkasowski. The ROCKOR stuff was mostly the work of Katja Gotthard, and some work was from Arjan Kortholt. And all the movies you have seen are from Ingrid Vetter, who runs my crystallography lab, really, and keeps everything going. And the RP2 story is from Stefan Weltl, who is now in Finland and does his postdoc there. I don't know why anybody would go to Finland, but he loves it there. And this is the collaboration with Peter von Hastert, who brought to us these ROCKOR kinases, these ROCKOR proteins. And since Jim mentioned being a hardcore football fan, this is the skyline of Dortmund, by the way. So this is a Ruhr area, very industrial area, and this was the last steel mill 10 years ago, or 2 years ago. It was dismantled piece by piece and brought to China, and so we don't have any steel or coal mines anymore, and the only thing that we still have is this football stadium here, and this is my team. This is where I'm going when I'm not on the road. Thank you very much.
Come up to the microphones, please. Well, let me start. Fred, really enjoyed that talk. The ROCKOR interaction's really intriguing. So is there a counterpart of the glutamine and ARG finger in that's tied into the dimer interface? And the second part of the question is, you made an analogy to RAF in terms of controlling the autophosphorylation, but since it's dimerization-based regulation, wouldn't there be a good analogy also to the dimerization-activated growth factor receptors, EGF receptors?
I think that's a much better analogy. So the first part of the question is the mechanism of all these dimerizing G proteins is totally different from those of RAS and RAS-CAP and the GAPs. They have many different ways of stimulating hydrolysis by the two active sites coming together. Very often it's a general base mechanism. There are glutamic acids involved and so on, it's just the localization of the metal ions that are involved. But the mechanism in these dimerizing GTPases is totally different. The second question is the analogy to dimerizing? Yeah, that's the wonderful sort of analogy that they have to dimerize and transphosphorylate each other, just as these guys. We don't know the structure of the rest of the protein. Obviously, it would be wonderful to have that, but it's obviously a model that we think they are inactive because they can't come together, but by GTP binding, there is a conformation change that induces these kinase domains to come together and then activate each other.
Well, let's talk. So two questions for you. First question is, you shown several mutations in the RAS gap, and specifically talk about arginine finger mutations, and those mutations are associated with the disease. So I was wondering whether the RAS or RAP mutation, activity in those patients are upregulated.
Yeah, clearly. I mean, in RAS, you mean the neurofibromin mutation?
Yes.
Yeah, people have shown that in certain cell types, schwannomas, for example, you find a high level of RAS GTP.
I see.
So I mean, there is still a mystery around the whole story in the sense that there are 11 or 13 RAS gaps, and you mutate one, and you find an increased level of RAS GTP. That is sort of a big mystery that nobody understands. Why the other 10 or 11 or whatever RAS gaps don't take over. It probably has to do with the special activity of neurofibromin, which has the highest intrinsic or the highest, or let's say the highest activity probably in the cytoplasm. But still, there are problems associated. We know there is an increased level of RAS GTP in most of these tumors. That has been shown. But what is not clear is, first of all, why not the other RAS gaps take over? Second, why it is a disease that is confined to neural crest-derived cells. It's really schwannomas, fibromas, and so on. Why is it not found in many other tumors? You really find this disease, so people are born with that mutation, but yet they have only tumors of neural crest-derived cells. Right. Again, that is not understood. And right. Neurofibromin is normally expressed everywhere.
Okay. All right. So second question is, I'm kind of remember you just mentioned that people probably around in this field try to find a way to allow the RAS GTPase defective form to hydrolyze GTP. That's a great idea, I think. So do you have any idea?
That's what everybody else tells us. Yeah.
Yeah. Do you have an idea you can share with us right now? So any approach or anything you're trying to do?
Yeah. We're trying many approaches and have had a lot of failures, I must say.
Right, but because this is going to be important. I mean, many cancers.
My feeling is, I mean, obviously you never know about toxicity and so on, but it would be the most direct way of treating RAS-derived tumors. If you know only in the end, but if these tumors are driven by RAS, which some tumors have a RAS mutation, but they're not driven by RAS, but other, let's say, pancreatic tumors are clearly driven by oncogenic RAS. So this would be the most direct way to treat them. It would also be a direct way to treat neurofibromas, by the way. So the problem is not the chemistry of this, because this RAP-GAP story shows you that all you need to do is take a helix. It's an asparagine there that pokes into the active site, stabilize the water. That is an effect of 10 to the 5-fold. Which is obviously, it takes a whole protein to correctly position that helix there. But the chemistry, I'm saying, is fairly straightforward. You need stabilize the water or maybe bring a general base into there, and it would work.
So do you think you need a RAS gap to get involved to stabilize the gamma phosphate to allow water molecule to attack?
Yeah, but obviously people have asked us, why don't you modify RAS-GAP so that it can work on oncogenic RAS? I mean, it would obviously be an approach. it's kind of difficult because there is not much room in the complex. There's not much room for anything. But it wouldn't be a good drug target. I mean, a good drug lead compound. I mean, a whole protein is not something that you would go for. We are going for small molecules, obviously.
Thank you so much. That's a great idea.
I have a question for the LARC kinase one. So if you make a kinase inactive, does it affect the GTPase, or if you, because I would think that GTPase reaction rates have to be very finely tuned to the kinase, so you have enough time for the kinase domain to hang out next to each other to transphosphorylate. Yeah. But is there actually also communication back from the kinase to the GTPase?
Reciprocal. I mean, the problem is, in the whole LARC kinase field is that you cannot, I mean, we cannot make any of the human proteins to really look at that in very carefully. People isolated from mouse brain sort of tells you that it must be difficult to make, and then they get a microgram out. So at least I don't know any study that shows that. I mean, it's clearly, people have shown that it needs GTP binding for the function. They show that it needs a GTPase for function. But whether the kinase activity affects the GTPase reaction, I don't know of any studies. Like I say, we would love to make that kinase and then also solve the structure of the human protein, obviously, but it's absolutely impossible at this moment.
Okay, let's take one more question.
I was wondering if you can comment of a nucleotide-free form of some of these proteins, like for dynamine, for example. I know that it's been suggested that they have some function.
Yeah. So all the RAS-like proteins don't like to be nucleotide-free. I mean, you have to really be careful. Like RAS, you can do it, but you have to treat it really, really carefully. But many of the others, they really collapse immediately when you take the nucleotide out of them, and they never are nucleotide-free in the cell. They have either GDP-bound or GTP. The only time they are nucleotide-free is when they bound to the exchange factor. But this is a very transient, very fast process. So those proteins which have so there are actually two classes. The high affinity proteins, they have sub-nanomolar to picomolar affinity. They never like to be free of nucleotide. All the other guys, especially the ones that dimerize, and dynamine is another case, they have a micromolar affinity. They don't need any exchange factor, so they can exchange, but they can also be freed of nucleotide, and then it's fine. So many of, at least the ones that I have worked with, that are dimerizing, they are fine when you take nucleotide away. Remember, so there are these two kinds. And strangely enough, it's really almost micromolar for all the other guys and all sub-nanomolar for all the others, more or less. I mean, all the examples that I know, at least. So they don't, to put a short answer to you is, the other one don't like nucleotide-free, these ones do like to be nucleotide-free. Whether that is a functional state is a different story, obviously, because at millimolar concentration of GTP in the cell, they never, and even dynamine will never be nucleotide-free either, right? Or let's say it's 0.5 millimolar GTP, but even at micromolar, it will be totally saturated.
So you don't think that, like, there is a third part to this cycle that will be a nucleotide-free and will be a functional?
Unlikely, because, I mean...
No, I agree.
If you consider the concentrations in the cell, it doesn't make any sense that they would ever be nucleotide-free. But they still, in vitro, you can take it away and they're happy.
Yeah.
Okay.
Okay, let's thank Fred and continue the discussion over cookies.